Hello, everyone. Welcome to the 8th NeuroTechX webinar. My name is Sydney Swain-Simon, and I'm one of the co-founders of NeuroTechX. If this is your first time hearing about our organization, we are an international community that facilitates the advancement of neurotechnology. We have a focus on open and accessible neurotechnology, and our community has helped to innovate in the domain through a variety of different projects. We have had contact points with over 11,000 people worldwide and have done collectively over 150 events. We also have local chapters where you can meet others. Because we have such a large international network, the goal of these webinars is to give people from different countries and different disciplines the chance to learn about different related subjects. We will be doing webinars on different themes, as well as showcase some of the amazing projects and research that our community does. We are always looking to improve the webinars, so please let us know how we could do a better job. With that in mind, we have two things you should know before we get started. Number one, please be respectful to those in chat. And number two, if you have any questions, post them in chat. I'll be monitoring it. I'll be monitoring it on my side. If you want to ask a question, just write question before typing it. The final thing before we begin is that if you're interested in doing your own NeuroTechX webinar, please send me a message on the NeuroTechX Slack or email me at sydney@neurotechx.com. With that, let's get started. For today, we have Jean Rintoul, who will be presenting Spectra, an open source biomedical imaging technology. Biomedical imaging has previously been expensive and near impossible to hack and experiment with. If more people experimented and understood how imaging works, we would move it forward much faster and make it these transformative technologies available to everyone. In this webinar, we'll talk about Spectra, a tiny 2-inch device that uses safe levels of AC current to recreate an image of any conductive material, such as your lungs, arms, or head, using the same tomographic reconstruction techniques as CAT scan. The implications of having a cheap imaging tool are huge across developing countries and could also be used in the realm of neuroimaging. If you have never met Jean, she's a super awesome person. She hopes to improve the pace of innovation in healthcare by pushing towards health technology commons. Previous experiences include bringing numerous consumer electronic biosensor products to market, from the Emotiv BCI to the Basis watch and Kiddo biosensor watch. After being published for her work in Cognitive Neuroscience and videogames training in Nature. So really cool profile. Thank you so much for joining us, Jean. Please take it away.
No problems. Hi. Yeah. So I'm going to talk about Spectra today. Got it right here, which is a 2-inch square biomedical imaging device. And thanks, Sydney. It was a pretty good introduction. And just to switch to the screen, can you remind me how to do that? I just...
Yeah, you should see a little button, I believe, on the left-hand side. You should see screen share.
Oh, great. Thank you. Okay. Screen sharing. There we go. Is that good?
Looks great.
Great. Wonderful. Yeah, so I'm going to give a little talk today about Spectra, this open source biomedical imaging project that I've been working on for a little while now. The goal is to create a gateway, a platform to move medical imaging forward, and to show people that it's actually, I wouldn't say easy, but you can hack on it. Anybody can hack on it and improve it. It doesn't have to be in the realms of a multi-million-dollar CAT scan or MRI for you to even go near it. You might ask, why make it accessible to people? If you remember, the democratization of the personal computer made a massive difference in how many people became programmers. So once upon a time, computers were the size of rooms, very exciting. A few people worked on it in labs, and that's wonderful. But what would be even better is if we can move that into more people's hands and have more people work on it and have more people become programmers, except for medical imaging. So right now, MRI is really big on the bottom left, but you've all seen Star Trek and the tricorder. Wouldn't it be wonderful if we could do that with a really tiny device? So that's the goal, to put that into people's hands and have them be able to experiment and improve with it. The vision would be like if everybody could access medical imaging and use it preventatively and more frequently. Say you could stick it in your Walgreens, hopefully not exactly like Theranos, but you could go in for a yearly test and you'll just like scan your whole body, and then you'd come in next year and they'll tell you whether anything had changed or not, whether you should be worried about anything. If there was something that was, of concern, you should definitely get more imaging done and go to a doctor. But just to get that sort of preliminary testing done would be massively life-changing for a lot of people. Also, considering the developing world have no access to medical imaging, to make it more accessible is huge. Coming back to the United States, it's the operating room hasn't really changed a lot in 30 years, and we spend so much on healthcare. The question is what's going on, and there's a lot of theories about what's going on. But it seems like there's some sort of problem occurring with the way that the innovation pipeline's working. So here on the flip side, we're going to look at an open source project. I'm going to start with a biomedical imaging overview of what's already out there, so you get an idea of what's good and what's bad and what the limits are. Then I'm going to introduce you to this technique, which is electrical impedance tomography, which is a kind of R&D technique that's done in labs. There's totally a lot of studies on it, and you can look them up, but I'm going to introduce it and then talk about the Open EIT project, and go to some sort of easy applications in the developing world, and then talk about possible next steps for the project. And I hope that at the end you can chime in with questions and ask whatever you want. So right now, medical imaging, there's an MRI, there's a CAT scanner. CAT scanner's got x-rays that kind of shine through your body from different angles. Ultrasound, using sort of high frequency sound waves. And EEG, which isn't exactly an imaging technique, but it's a very important technique nonetheless, and I want to include it because it relates to the neuroscience side of things. So when we sort of look at a biomedical imaging device, there's a few different ways we can sort of rank it. So there's different types of resolution, not just spatial resolution. There's contrast resolution as well, which is looking at the difference between different tissues. So for instance, if you want to look at the difference between soft tissues, an MRI is really good for that, but a CAT scan is really bad at that. But a CAT scan can see bones. So the contrast resolution is really important when you come to thinking about which particular imaging modality you should use to figure out a particular problem that a person's having. And time resolution is also an important one. So some things in the body happen over shorter or longer time scales. A typical example would be MRI versus EEG. An MRI looks at hemodynamic changes, so changes in blood flow. Now, that happens sort of over a curve that goes for about 10 seconds. So, it comes up over about 5 seconds and then decreases over the next 5. So you can't see really fast electrical changes, say. Now with EEG, you can see that because it has really amazing time resolution. But on the flip side, EEG doesn't have very good spatial resolution because it's like that sort of a couple of sensors in a soccer field issue of the inverse problem. So cost-wise, super expensive. MRIs are big as well. You definitely can't play with one on your desk. CAT scans, a couple million. Another thing to keep an eye out for is safety issues of using them. I'll just skip to this slide. So CAT scans, because they use x-rays, are ionizing radiation. And apparently Americans get most of their have the highest medical radiation in the world because they use so many CAT scans. So it's actually really bad for you to get a CAT scan. Don't get one unless you really need one, in which case you're probably almost, something is very urgent that's happening. So it's definitely worth it because you are saving your life. Just a little sort of point out about EEG. EEG is awesome and a good research tool, but it would be wonderful to move further and overcome its shortcomings. So a couple of things about what it doesn't measure. It doesn't actually measure action potentials. That's what you would like to measure, sort of a current source localization of when the nerve cell opens and the calcium ions come out of it. So it's also doesn't have very good spatial specificity, which is a massive problem. That's the inverse problem again. And it's actually only measuring signals perpendicular to the sensor and near the cortex as well. So there's a lot of issues with that. If we could get through that and get like a more accurately scaled and spatially localized signal, we would be able to understand a lot more about the fast electrical activity that's going on in the brain. So back to this sort of contrast and spatial and time resolution discussion. So different spatial resolution or time resolution will be good for different things that are wrong with you. So a chronic respiratory illness can be diagnosed with pretty poor spatial resolution, say pneumonia, or pulmonary edema, which is water on the lungs. So you need sort of medium time resolution, so to measure breathing, and pretty low spatial resolution. So I just want to point out that you don't need high spatial resolution to still be a useful medical imaging tool, because that's going to be highly relevant when we talk about electrical impedance tomography. So EIT, what is it? You might not have heard of it. It's a different technique. It's an active technique for one, compared to EEG. What it does is it sends small AC current through the conductive medium, and the current flows around different paths dependent on the frequency. To recreate an image, what you do on the bottom left here, you can see what they call a phantom, which is a typical test setup in any biomedical imaging field. They call it a phantom, which I think is a great word. And then what you do is you send the signals through, and through every combination of electrodes, and then you can apply a set of equations to recreate an image. And what you can get is what you see on the right is a kind of electrical impedance map. And this map on the right is at a single frequency, just at 10 kilohertz. And you actually get slightly different spatial maps at different frequencies, which is another interesting property that you can exploit. So that's a very brief explanation of electrical impedance tomography. And it's kind of cool that you can do imaging in any way with AC currents, because they don't rely on an expensive transducer like ultrasound. It's a very accessible technique. It would be easy to mass produce. It's currently being used in lung volume measurements, particularly on little babies in the NICU who are have lung issues. You can measure things like muscle, fat mass, gestural recognition, bladder, stomach fullness, breast and kidney cancer. There's a whole bunch of studies on that. Hemorrhage detection would be a great one, and depth of anesthesia via action potentials is another area where there's a bit of research on, which is sort of interesting, and I'll get to that in a bit. What's so good about it? It's non-invasive. It's much safer than X-rays and CAT scans. It's really tiny and compact. I just sort of showed you a little 2-inch square PCB. Inexpensive. It's got better source localization than EEG, and it's got good time resolution too. So it's pretty good for functional imaging. Disadvantages, not many people know about it. Big disadvantage. Spatial resolution is limited by the number of electrodes used, and also the difficulty of the inverse problem, which is a massive, inverse problems are a massive area, and mapping currents is quite tricky. And I'll sort of discuss the algorithms that are used in a little bit. It's unlikely to be able to acquire as high spatial resolution as MRI. But let's just have a look at this. The first MRI scan in 1977 is up there on the top left. That's a human thorax, or ribs and lungs. And it's really pretty bad. And down there on the bottom left, that's actually my head when I was at Ghazali Lab and put through an MRI scanner. And obviously, it's improved a lot. That's just a couple of years ago. On the right, we've got an early EIT scan. You can see the lungs and the heart in the middle. And the big question is what's next and how far can we push this technology? Can we push it as far as the MRI scan? I don't know. So that brings me to the Open EIT project and the idea that sometimes when we share information, we can move things forward faster. So here it is. You can check it out on GitHub. That's the address, and everything's open. It's just there. It's actually got a set of instructions at the top, which I encourage you to go to and sort of get a brief overview of what the project does. So just to discuss the algorithms for a second and how you actually reconstruct an image. Well, what you do is you send, as I mentioned, you send AC currents through at different angles, and they will go through the medium and produce a different impedance at the other side. And it's that impedance measurement and that change from output to input that you're measuring. You do that around in a circle, you create something called a sinogram, which is over here on the right. So we've rotated around the same way as a CAT scan and created a sinogram. And then we do an inverse radon transform. And bam, we recreate an image, and that technique is called back projection. Now, there are other techniques as well that use a finite element model, such as the Gauss-Newton method, or another method called the Graz consensus method, which are used in electrical impedance tomography. In the Open EIT project, I implement all three of those. They each have various pros and cons. For instance, Graz consensus is optimized to get higher spatial resolution in the middle and not around the edge to sort of overcome some of the contact impedance problems. And Gauss-Newton is your typical finite element model reconstruction, and back projection is the same technique as the CAT scan. If you're wondering whether sending little AC current through your body is safe, it is, as long as they're within the guidelines for safe use on humans, which is less than 10 microamps. And that's a set of standards called IEC 60601-1 guidelines. So just to put it in perspective, you know those sort of electrical ab stimulators that they advertise on late night TV? They're a few milliamps. So this is less than 10 microamps. So very tiny current is applied, absolutely within the safety standards. So this is what it looks like. This is the Open EIT project. I call the device itself Spectra, because at different frequencies it gets a dielectric spectrum, and I think that's one of the most exciting parts about the information that it gets, which is ideal for it's this big dimensional data set, which would be ideal for machine learning. So basically it's got 32 electrode, Bluetooth, accelerometer, pretty good sample rates, 16-bit resolution of the analog to digital converter. You can do bioimpedance spectroscopy. It's really small, open source software, a bunch of algorithms. The frequency range that it runs between is 80 hertz to 80 kilohertz. It's safe, and it comes with the cylindrical tank, which means that you can do testing in the Phantom, which is the typical biomedical imaging test setup. So this is what it looks like when you run it. So here is the tank with 2 cups on the left, and you're able to localize where the cups are just using current. On the right, I move the cup around anti-clockwise, and you can see that the reconstructed image mirrors exactly where the cup is located. So that's showing that the algorithms and everything works. And you can put anything in that tank that you're interested in categorizing at all. And I'll show you a few of the variations of techniques that you can do with it. So this is what the software looks like. So you connect to the device, and you have 3 different choices of types of things you can do with it. You can get time series impedance data, which is down on the bottom right. So that's at 50 kilohertz, and I've attached it to my chest, and I'm measuring both my lung expansion and contraction, and my the blood moved through my heart valves. And what's cool about that is that it's not an ECG. It's a different kind of measurement. It's measuring the material change due to the dielectric materials, the impedance changing. So it's a mechanical change. So your heart has blood, the valves open and shut, and you're measuring that blood flow change. You want to see how much air you've taken into your lungs. It's measuring that change, which is quite different from just the sort of passive electrical signal. Up here on the right is the bioimpedance spectroscopy. What that is where you send a bunch of different frequencies through different objects. Here I've sent it through an apple and a sweet potato and water, and you get different signatures. So you can imagine doing this on a biological substrate as well, like a tumor versus healthy tissue, to get different spectrums. So this is the spectroscopy angle. If you want to differentiate any material, this is a great technique and should be exploited by people far more for a lot more applications. And then here you can just see the image reconstruction, and it just runs in real time. You can switch the number of electrodes to 8, 16, or 32. This is an early video. Let's have a look here of it working. So here I'm just moving a cup anti-clockwise, and you can see it mirroring it in the image. This is back when I just had the 8 electrodes set up. I've since improved the device and moved to 32 electrodes to get much better spatial resolution. So just showing you that working. How do I get out of here? And then we can also show you gestural control. Just a second. Oops. Just jump to a sort of later part in the video. So gestural control is another application where you do different arm expressions, and it's measuring the muscle changes in your arm to be able to recognize different gestures. Here you can see basically me doing the same gesture and different gestures, and the same pattern emerges on the reconstruction. Here we go. Lung imaging. You can wrap it around your thorax and do lung imaging. It is a sort of overall blurry image. That is the nature of electrical impedance tomography due to the difficulty of the inverse problem. But considering that it's a very small, portable biomedical imaging device, this is much more efficient than a CAT scan. So you can also do it continually and safely. So it has different advantages. So I've described this sort of lung contraction or lung expansion data, which is the time series data. And here is the bioimpedance spectroscopy. What I've done, you can see in the tank, I've got different fruits. And I've created a simple classifier, a very simple classifier from these spectrums. And what that lets me do is I'm able to spatially localize and identify where the apple or the sweet potato is, which I think is pretty exciting. What that's able to do is identify a material at a spatial location. So imagine that we replace the apple with a tumor or something else that we wanted to find, like a blood clot. Here's a few ideas on different applications. Lung imaging is obviously a big one. Pneumonia detection in developing countries, fruit ripeness, embryo and cell analysis, tumor detection, If you wanted tools for real-time feedback in surgery, it'd be pretty good. Monitoring of fat, muscle, bladder, stomach changes, industrial applications, material flow in pipes, or measuring ice, fetal breath monitoring. And over here on the right, I'm doing a deep breath and a shallow breath, and you can see the exact volume that I'm breathing in with my lungs. And I think that's also pretty cool that you can just see that like that. So I think the moral of the story is that biomedical imaging shouldn't be seen as something that's really hard and impossible for people to hack with and experiment with. And if we started to experiment with it together and create a commons in this area, a global commons would be great. We could move medical imaging forward and make it cheaper and more accessible for everyone much faster. So again, a reminder about what the first MRI scan looked like and where we've come and where an early EIT scan is. And the question is, what's next? So what have we been able to do? What's the best result for EIT to date, you might ask? Well, it's actually pretty good. So the spatial resolution has got to 200 microns. This is in David Holder's lab at UCL in London, and a gentleman called Kiro Aristovich wrote a paper sort of describing this, and the time resolution less than 2 milliseconds, which is pretty exciting for measuring, say, action potentials. They did this using an ECOG array on rats, and they were able to localize where action potentials are originating from, which is super exciting. So with a basic biomedical imaging device that is cheap to mass produce and extremely portable and low power, we should remember that 2/3 of the world has no access at all to any kind of medical imaging. So we could actually do a lot of good with something that is just available and can do some basic things. So we go back to this image again and look at spatial resolution and time resolution, and we just want to pick something that could be useful in a developing nation. We could pick, say, pulmonary edema, water on the lungs, or a chronic respiratory illness like pneumonia, which is a big problem. Maybe tuberculosis would be good too. So here we see an EIT image on the bottom right of the lungs. And you can see that one lung is obstructed because it's much fainter. It's not expanding properly. You can see the time series measurement on the top right, and it's absolutely not expanding properly. You can even see sort of whereabouts the problem is occurring. So that's there's a lot of studies on this already, and you should check it out. But that's a great use for it right there. So now we've sort of talked about what EIT is, what the Open EIT project is. I'm sort of do a little recap on where it's at. So we've got a little device. It can do permittivity reconstructions, and you can do it in a tank, or you can wrap it around your body as it's IEC compliant, so it's safe. It's also got Bluetooth, so you can move around with it. So yeah, if you want to help out, go to openeit.github.io and have a look. I'm planning on doing a crowdfunding campaign soon. I'd love people's support with that. How can we improve it? How can we push it further? So if we have a look, obviously more electrodes can give you more spatial resolution. On the bottom right, I show how that works. Basically, you get sort of this exponential effect where you get from 8 to 16 to 32 electrodes. You could try different kinds of tomography with it. So I've just done AC current-based tomography, but because we're sending out controlled current source, you could actually use other things like perhaps infrared light and do diffuse optical tomography. Maybe you would like to get an immersion sound transducer and do acoustic tomography as well. All of these things work, and I think they really warrant more investigation. You can put it in a big magnet and you get some advantages as well. That's called MR-EIT. You put it. They're putting EIT in an MRI. There's some interesting work in acousto-electrical tomography coming out by Yexian Qin. I don't know exactly how to pronounce it, but he's using ultrasound and the piezoelectric effect to convert sound to electrical signals to measure current source localization in the brain at a precise point in space, which is pretty exciting. With the reconstruction, the algorithms, right now we make some really simplifying physics assumptions when we solve them. For instance, we pretend it's a resistor network, or the body is not a resistor network. It does have resistance, and it is a network of resistors, but it also has capacitive and inductive properties. And we should surely include those too, but that makes it all a little bit more complex. So perhaps for this machine learning approach would be quite good because we have such a complex data set. Improving the time resolution. As I mentioned before, there's some exciting studies showing that you can measure action potentials with this. So the way that works, if you're wondering, is that when the calcium ions are released from the nerve cell, the opening in the nerve cell creates a changing in impedance, which can then be measured. So the current goes through the cell instead of around it. So you can measure that to see when action potentials are occurring. So non-invasive electrophysiology would be great. So there's some interesting work by a gentleman called Nir Grossman, and he came out with a paper in 2017 on non-invasive deep brain stimulation via temporally interfering electric fields. So he's, in case you haven't picked this up yet, anything that you can read with, like EIT, at a different power level, you can also write with. And what he's doing is he's getting two current sources at different AC frequencies and offsetting their frequency just by a little bit, like I think it's at 2 and 2.04 kilohertz. When he combines them together, they create a beat frequency. And that beat frequency, which you can see in the middle on the image on the right, is a lower frequency signal. Say it's at like 10 hertz, which is something that the brain responds to. He's been able to show that he can stimulate the nerves at a precise location, non-invasively. So when he creates this big frequency setup, those nerve bundles at that location are stimulated in a coherent manner. So he's getting this 10 hertz echo from them. And I think that's very interesting and exciting, what we could do with deep, non-invasive deep brain stimulation. So there's all these wonderful things that we could do without the need for surgery, which would be wonderful for a lot of neurological problems. And then there's improvement of the algorithms using modifications and more intelligence in Maxwell's equations. So there's a lot of different kind of techniques in radar that I think could be moved into this area. So with the things we've been doing on a larger scale, like phased array and beamforming, should all be coming back into this sort of current steering methods to look inside the body, to both read and write. So those were some possible next steps and directions that it could go in. Obviously, you know, Spectra device is an open source effort, and I'd like to thank the different contributors Marianne LeBorn has helped with some of the software, and Ben Wan created a great library for EIT algorithms. Clement, who I met in Germany when I spoke at 34C3, gave me some wonderful hints on flex electrode arrays and so forth. So it's fantastic to sort of get hints, help, and feedback on how things are going and how it could be made better. And I welcome you to get involved in like whatever capacity you feel comfortable with. Even if it's just saying, oh, I've got an idea how you should do it better. If it's a good idea, great. It's nice to hear those things. So I want to sort of close up with a kind of story about how surgery has changed and remind you that we used to use saws and big knives, and the hairdresser and the surgeon were one and the same profession. And they became separate at some point in time. When you got an infected limb or needed a haircut, basically that same knife or saw would be used. Just cut off the infected body part. You'd definitely be thankful to still have your life if you survive the surgery. And we've really come a long way. Now we can do things like keyhole surgery, which is far less invasive. We have much better survival rates. However, the really big opening is non-invasive bioelectronics, in my opinion. So if we can both read and write in a really controlled manner, there's really no limit to what complex procedures that we can do in a highly focused and non-invasive way where we don't have to open people up anymore. So this would just be absolutely revolutionary for surgery and sort of health, our general health and well-being. So I see that as the future of medicine, to be able to do these highly focused medical procedures all non-invasively. So you could have a lot more sort of outpatient procedures where you just focused, like removed just the tumor and then somebody walked home an hour later. So this is the Open EIT project to look inside the body and to understand biomedical imaging and make it something that we can all experiment with and improve upon. If you want to join the mailing list, go to mindseyebiomedical.com. I'm currently trying to ramp up for like a crowdfunding campaign and make a bunch of these. You know, you could support this effort by buying a kit of one of these Spectra devices, which would be fantastic. And I'd love your feedback on everything. And yeah, please help us make a gateway to move medical imaging forward. It's really important. Okay, yeah, that's it. If anybody has any questions, I'm happy to answer them.
Thank you so much. So the first question we had from AJ in chat, he was curious to know what frequency do you measure the impedance at? And specifically, he's wondering what the frequency is of the signal that is being injected into the EIT.
So you can control the frequency of the signal. For the Spectra device, I'm injecting between 80 hertz, which is quite low, to 80 kilohertz. If you look at the bioimpedance spectroscopy plots that I popped up, then you can sort of see the range of frequencies that it can access. I picked those frequencies for a reason. It's because you can see some of the biggest changes in cells at those frequencies. So it is for those frequencies are selected for a physiological reason because they're the richest in information. Not to say that there might not be other information in other areas, just to say that that is typically what's used to get really good, they call it dielectric spectrums of soft tissue in humans. And for the images, like the lung image, I use a single frequency only, of 50 kilohertz. But you can change that to anything between 80 hertz and 80 kilohertz. And one of the kind of cool things about the technique is that you get kind of different images at the different frequencies. So you get a slightly different image if you just do it at 80 hertz of the lungs than you do at 80 kilohertz. So that's extra information that you could use. So just to put that out there.
Another question from chat. Javier Serrano is curious to know what is the price of the kit, and if you have a price set just yet.
TBD. So I'm currently working to reduce the price of the so it's got a bunch of kind of precision parts, not to mention the main microcontroller, and I have some really nice multiplexers on there, and I'm sort of able to get a pretty good deal now, and it's going to depend a little bit on the so basically I can make it cheaper the more that I sell. So if I can get a big group order on crowd supply soon, then I can make it much cheaper. So I'm trying to make it as cheap as possible is the answer now. And if you want to get one that's not as cheap as what will be available on crowd supply, you can email me privately, and I can sell you a functional device now, but it will not be quite as cheap as what I will be able to do on crowd supply, just to put that out there.
Great. So a question I had actually was, you mentioned that in the current design you have 32 electrodes in the system. Is there ways of expanding it to go beyond it, or have you found that 32 is the maximum that you can achieve?
No, you could go to any number you want. So at the end of the day, it would be a bit of a trade-off with time, how quickly you want to get the image, because it is multiplexing. So that's the trade-off. So if it's something where you're happy to get just a static image, you could totally. So it has a little patch-out adapter, and you could patch it out to even more multiplexers. There's no problem with doing that, and basically pick any number of electrodes you want. I just sort of thought, 32 is a good place. You can easily just in the software switch it down to doing an 8 electrode image or a 16 electrode image, which does give you a faster time resolution. So to switch to say 128 electrodes, that would be you would have to patch it out to a couple more multiplexers to do that. Yeah.
Great. So 2 more questions from chat. So AJ wants to know what do you need to get this product to market faster, and Javier Serrano with a similar question, but he's curious to know, do you have an estimated date for the crowdfunding?
So yeah, that's a really good question. So obviously I've got everything pretty sort of honed in on sort of the prototype. I've started making the functional test programmer, in fact, and I can program the devices, and but I haven't fully finished that. So that means that every device will be tested for functionality before it reaches anybody's hands, which is always nice. So crowd supply is actually in the process of video editing. So we've done the video shoot, and it's in a video editing process, which hopefully is a week or 2, and I'm really eager to just have it go live. So hopefully, I'm ready to go. In a few weeks' time, hopefully we will be launched, and I would love people's support on that to keep this project going. And I'd love to get the kit into your hands and see what you come up with, see what you do with it. And get your feedback on it. And I'm super curious to see, what are you measuring? Do you want to measure breathing? Do you want to the cell analysis is a really interesting other option. So there's just a lot of different sort of physiological applications that you can experiment with this. And I just think it's a great technology which should be in more people's hands, and more people should know about it.
And are there ways that the community can help to get you there quicker?
That's a good question. I don't have I can't think of any. So short of saying, hey, crowd supply, go faster. So it's not exactly fully in my hands because I have sort of outsourced part of it to them. But I'm ready to go, and I'm ready to move as fast as possible. And I will endeavor to do that. I can't think of anything off the top of my head, but yeah, it is almost ready. I do have like fully functional, nice prototypes. I've even got tutorials at the Open EIT.github.io address, and I can't wait for people to try them and then say, "Oh, you missed out this vital piece of information," and I will happily update it. But hopefully, I haven't. But yes.
Awesome. So a few questions on my side. You mentioned that there's a possibility of a right functionality or a stimulation functionality with the EIT. I don't know if you actually mentioned it, but is there a level? What do you think is what is the level of spatial resolution that is feasible with it? How in depth can you go in terms of stimulation?
You mean how deep into soft tissue or in the brain? So with the stimulation, so I'm familiar with the. So so the device Spectra is set so that it is within the safe limits, but it comes with little header pins to do other tests with, and you can just plug those pins, and then you can change the current to any level you want. So I've made it so that it is by default safe. And then if you want to do other things, obviously that's your own business. And there's I can sort of describe how you can change the current level. And then it has a DDS, digital synthesis, so you can create any waveform you want and experiment with that. Yeah, so I would have to look up the Neil Grossman paper to look at exactly the levels, but this could generate anything up to 5 milliamps, which is quite high. You will never need to go that high. And that would be a great thing to test in like a phantom, in the phantom test setup is a good place to start with the kinds of current variation experiments, because it's one, it's safe. And if you can put little EEG sensors in there, you can sort of measure what you're sending out quite exactly. And then you can have a look at whether you're creating the physiological effect or not in perhaps an animal or a squid or something else. But yes.
So, but would it be something at the level of stimulation like TDCS or like functional ultrasound? Is it just like at the highest like cortical level, or is it going in depth a little bit more?
Oh, okay, got it. Yeah, it actually goes in depth. So this is one of the other super awesome things about it. The current goes all the way through. By that I mean all the way through from one side to the other side. That's basically, I mean, that's how it works. So you are getting in-depth info, which is why they've done a whole bunch of studies on anesthesiology, because that's looking at sort of deeper areas in your brain. And what is their electrical activity to make sure that you're actually properly unconscious. So EEG sort of has some trouble with doing that. So this actually goes all the way through, so you can access. There's no limit on the depth except for the power. As long as you can sense the signal at the other side, then you're good. If you can't, so say it's an elephant or something very large, you might need to turn the power up a little bit. So you can always vary the current output to be able to sort of measure a larger object or a smaller object. But as long as you can measure on the other side, you are measuring it at the object at its deepest point, at the midpoint. I hope that answers the question.
Well, you're talking more about the imaging side, right?
Yeah, on the imaging side.
I was focusing more on the stimulation side, because you had the one slide discussing that there is the feasibility of doing stimulation with it, right? Or did I misunderstand?
So I was sort of citing a Neil Grossman paper and his work. I haven't done that. I've done imaging, time series impedance measures, and bioimpedance spectroscopy. So I've focused on the read. The write you could absolutely get into, but I haven't. And I like you can create your own waveforms and do that. But I haven't personally focused on that side. I did do a tiny little bit of computational simulation of his work. which is something that I would encourage anybody who wants to get into the current stimulation area to do first. It's so you can just start by doing a little simulation of Maxwell's equations, time varying equations, which are a bit more complex than the static equations, and then you can sort of simulate the signal through the medium, and then you can do it in a phantom, then you can move to biological organism. So that would be the sort of development pathway that I would sort of vaguely, I would suggest for doing current stimulation. But yes, you could try it, but I haven't done that one yet.
Okay, so maybe one or two more questions before we wrap up here. You were mentioning that you were using the analogy that this technology is kind of at the early stages of the MRI, where the resolution is still rather limited, but with time, we can get much better idea of what's happening, more in depth, knowledge and better mapping completed. What are some of the technical challenges that you think need to be overcome in order to be able to get to that level? Like from your perspective of working on this, where are the limitations right now?
Right. Yeah. So one thing that I'm sort of excited about is the hybrid modality. So you can do tomography with anything that can measure an impedance. And so EIT is super interesting in that you can get spatial localization non-invasively. And I wonder if there's a way to focus it more, because the limitation seems to be the spatial resolution, and everybody just seems to look at spatial resolution first, even though it gets really great contrast and time resolution. So I would like to sort of, if we wanted to improve the spatial resolution, I would suggest a hybrid modality. The most exciting paper that I read recently on that, which I sort of alluded to in the talk, was 4D acousto-electric imaging of current sources in a human head phantom, which uses the piezoelectric effect to basically do EIT with an ultrasound transducer at a precise localized point in space. And I think that works very interesting. And that has potential to get higher spatial resolution, but there might be other approaches as well. The other main area, there's a new algorithm that's come out in EIT, which I neglected to mention, called DBAR. And there's been some awesome reconstruction results with that. I haven't personally implemented DBAR, but there are implementations out there. And I think that algorithm seems to solve the spatial localization problem somewhat better than the existing ones, but it's quite new as well. That would be an enhancement to go down. Yeah.
I don't know if Eamon's question is directed to you or someone in chat, but he wrote, aren't the frequencies low enough that Maxwell's equations don't really come into play? Not sure of the context, but maybe you understand what he's talking about?
So Maxwell's equations kind of always come into play. It just depends what version of Maxwell's equations. So for this, I'm making the inaccurate assumption that they're static and not a time-varying field. And so if you wanted to enhance the results, I would suggest using you could use time-varying Maxwell's equations, but there's all sorts of different ways to formulate the basic equations of Maxwell's equations. And a lot of them just make a whole bunch of simplifications to make it easier to solve. And you're welcome to check out the PyEIT library, which you can see at openeit.github.io, which has the equations and the algorithms written down to see what has been done so far. But yeah, they do make they make some simplifying assumptions, and it would be better to not make those assumptions, because basically based on Neil Grossman's work, it is a time-varying version of Maxwell's equations, and we should be applying that instead.
And we'll give the final question to Javier here. So what is the duration of the battery? And so he's curious to know, if it could be a mobile accessory and something that could be worn in, wearable technology, maybe integrated in a shirt or something similar.
Absolutely, yeah. So it depends what frequency and how often you're collecting the signal, obviously, and what firmware protocol you're running. So it's a very low-powered MCU based on the ADU CM350, and So, and it also depends how often you want to send the info out via Bluetooth, because it does have Bluetooth. So obviously the time duration, the battery has a battery on it. It depends on how you set that up. So I haven't firmware optimized it. I've got it sort of so that you can easily switch between the different modes and do different measurements. And yeah, so you could firmware optimize it. But the power, it's a very power optimized chip, to answer your question.
Great. Thank you so much, Jean, for presenting today. It was really, really fascinating subject, and really, really excited that you're able to share it with us. And thank you for everyone else for being able to join us today as well. If you did like this stream, please subscribe to be informed about future videos. If you'd like to get a monthly dose of what is happening in the neurotech field, please subscribe to our newsletter. The link can be found in the video description. With that, we hope you have a good day, and we'll see you next time.